Assalamu alaikum students. I hope everyone will be fine at home. Um, Previously, we talked about the light trapping, trapping light energy. Then we had a look on chromatography and how we can separate the photosynthetic pigments with the help of paper chromatography. Then their RF can be calculated, etc. Today we are going to discuss about the two subtopics, and they are how we can grow the plants, not how we can grow, like what if plants are grown in the protected environment, for example glass houses. And the second is C4 plants. What are the C4 plants? Why are they called so? What are their adaptations to the particular environment in which they are growing? And what is the advantage of being a C4 plant? So first of all, we will start with growing plants in protected environment. Now we know that obviously when a plant is growing, and obviously then it's going to produce the fruits, and obviously if we are talking about the crop plants, then the crop yield is there. So this food production actually depends on photosynthesis. Okay, so and we know that the photosynthesis is affected by many factors. For example, we have already discussed that light intensity, wavelength, temperature, carbon dioxide concentration, etc. Now you see, when we say food production depends on photosynthesis, and we know that the rate of photosynthesis is affected by many environmental factors, then obviously we can understand that when the crops are growing in their natural condition, it's not easy to control the environment of those crops in the natural condition. Obviously we cannot control those environmental factors as per our own choice when the crop plants, they are growing in the natural conditions. However, when we are talking about the growth of plants in the protected environments, that is a different method. In protected environment, for example a glass house, it's much easier to control the various environmental factors there. And as per that, we can obviously, if we are able to manage the different factors in that surroundings, that's going to have effect on the rate of photosynthesis. More rate of photosynthesis would be there, so more crop yield will be there, right? So plants when grown in the protected environment, that is a different method. And obviously this is because, once again as I said, this is because of our understanding of effect of various environmental factors on the rate of photosynthesis. And obviously this understanding enables us to manage the growth of crops when grown in protected environments such as glass houses. Okay, and main aim of growing the crop plants in protected environment, that is to increase the yield of the crop. And obviously one more thing, that now we see many vegetables, fruits, they are available even in those seasons which are not their particular season, like bhindiyan, karele, now we see in the winter season as well. So this is because they such vegetables, they require certain temperature, certain other factors to be controlled. And in glass houses, if we can change those factors, we are able to grow the crop plants in out of season as well. But basically the main aim is behind this all is to increase the yield of the crop. Okay, like one example given over here is there as well, that many hectares of tomato plants, they're grown in the greenhouses. Okay, and now what happened in glass houses, sorry. And what happens inside the glass houses? Actually, there are very sophisticated sensors, right? And those sensors, they continuously monitor the light intensity, the various environmental factors. For example, the light intensity, humidity of atmosphere, concentration of carbon dioxide, temperature, etc. Second thing, now these are the main, these are the examples of those environmental factors which are controlled inside the glass houses. And this monitoring is done by sensors. Actually, everything inside the glass houses, that is digitally controlled with the help of computers. Now you see one more thing. Plants, they grow hydroponically. Now what does mean the hydroponic growth? Hydroponically means that their roots are actually growing in a nutrient solution. Okay, and that nutrient solution, obviously nutrient solution means various nutrients are added. So one advantage of this is that the content of the nutrients, it can be varied at different stages of plant growth as per requirement, like where we have to add more magnesium, where we have to add more potassium, etc. Okay, we know that all these things are required for the growth of the plant. So basically hydroponically means that the roots of the plants, they're growing in a nutrient solution, and the contents of the nutrient solution can be varied at different stages of plant's growth. Now you can see this is a picture of one of the glass house and you can see water here. So this is the nutrient solution as well. Now, all these factors, as I told earlier, that they are managed digitally with the help of a computer, and that is once again to maximize the yield of the crops. Okay, okay. One more advantage of growing crops in the glass houses is that, and that is one very important advantage as well, that you see there are different, just like we suffer from many diseases, in the same way the plants, they are also affected by different diseases. They can be fungal diseases, they are mostly the fungal and the pest diseases. Okay, so different fungal and the pest diseases, they are more easily controlled inside the glass houses than when the plants are grown in the fields, in the natural condition in the fields. So once again, this point is going to further increase the yield. Okay, so this was all about, it was a very small subtopic. So this was all about that what are the advantages of growing crop plants, particularly in the protected environment, for example glass houses, how different environments are controlled, how different factors are controlled over there. That's it. Okay, now we come to the C4 plants. Now before coming to the C4 plants, we are going, actually there are three categories of plants according to the photosynthesis. It's C3 plants, it's C4 plants, and it's CAN plants. Okay, but CAN plants, they are not in the syllabus at all. Okay, it's generally the C3 plants and the C4 plants. C3 plants we have already discussed when we discussed the process of photosynthesis. Now we will see how we have discussed this actually. Okay, actually, if you remember, in light independent stage of photosynthesis, which was basically only Calvin cycle, we know that. If you remember that carbon dioxide combines with ribulose bisphosphate. Okay, and we know that carbon dioxide obviously contains one carbon. Ribulose bisphosphate was a 5 carbon molecule. So as a result, the molecule or the compound which is formed, that was 6 carbon compound. But we know that this 6 carbon compound, it is highly unstable. So as soon as it is formed, it immediately breaks up or it splits to form two molecules of 3 carbon. Okay, so that's why when we are talking about the Calvin cycle, as far as the number of carbon is concerned, the very first compound to be considered there is this 3 carbon compound, and that was phosphoenol that was sorry, glycerate 3 phosphate. So basically, as we say, so the very first compound formed in light independent stage of photosynthesis is a 3 carbon compound. So plants, they are called as C3 plants. Okay, so just remember that this C3 or the C4 terminology is with reference to the compound, the very first compound formed in the light independent stage of photosynthesis, whether it has 3 carbon or whether it has 4 carbons. Okay, so in those plants where the very first compound formed, that is the 3 carbon compound, those plants are called as C3 plants, like rice plants are there example. So I hope this is now clear what is meant by C3, and you can guess from that what can be a C4 plant. Okay, now this is, you see, these are the C4 plants. There are various examples of C4 plants, like sorghum is there, millet is there, maize is there, sugarcane is there. These are the actually these are the four most common crop plants in the which are included in the category of C4 plants. Now, once again, maize, sorghum, most other tropical grasses. Okay. Now, in these, mostly you will see that these plants are growing in the tropical parts of the world. Okay. An example is maize, sorghum, and millet, sugarcane, right? So this you can see on the right side is the picture of the sorghum plants. Okay. sorghum आई थिंक उर्दू में कहते हैं जवार को, ठीक है, और जो मिलेट है ना उसको हम कहते हैं बाजरा, right? But you see, in our countries, bajra we are going to give it to the parrots and the pigeons, etc. But in many parts of the world, like many African countries, the millet and the sorghum, they are the main staple food of the people over there. The staple food, the main food which is taken, the main crop which is taken as which forms the basic food of the people of that country, of that region, that is called as the staple food. So though in our country, because in our country we have the wheat, but in certain parts of the world like African countries, sorghum and the millet, they are used as the staple food as well. And maize, maize corn, that is used in Europe a lot. They make cakes, it's used a lot in bakeries, while we have comparatively lesser use of corn in our country. So maize, sorghum, once again millet, sugarcane, and many other tropical grasses, situation is different. Something different occurs during the process of photosynthesis, particularly when we are talking about the light independent reaction. Okay, so in light independent reaction, the very first compound formed, that is a 4-carbon compound. So that's why these plants are called as C4 plants. Okay, so C3 plants कौन से हैं जिनमें in those plants where in light independent stage the very first compound formed, if it is a 3-carbon compound, those plants are called C3 plants. If it is a 4-carbon compound, then obviously these plants are called as C4 plants. Okay, for example, as once again this is the sorghum plant. Now, what is the problem with the C4 plants? Why there is a why there is a difference in the light independent stage in C4 plants as compared to C3 plants? Okay. Now we know that when carbon dioxide combines with ribulose bisphosphate, this reaction is catalyzed by this enzyme which is called as Rubisco. Okay. Now there is one problem with Rubisco. Rubisco, if it it catalyzes the combination of carbon dioxide with ribulose bisphosphate, but at the same time it can also catalyze the reaction of oxygen with ribulose bisphosphate. Okay, the main substrate remains the same, means ribulose bisphosphate, but Rubisco can catalyze the reaction of carbon dioxide with ribulose bisphosphate, and at the same time it can catalyze the reaction of oxygen with ribulose bisphosphate. You see, when oxygen combines with ribulose bisphosphate in the presence of Rubisco, this process is called as this reaction is called as photorespiration. Why photorespiration? Photo, obviously, in the presence of light is there, and respiration, obviously, because in respiration also oxygen is utilized, so that's why oxygen is going to be the here, so that's why this is called as photorespiration. Now you can understand, if Rubisco catalyzes the reaction of oxygen with ribulose bisphosphate. So let's say some of the ribulose bisphosphate has combined with oxygen. So don't you think so there will be less photosynthesis taking place there, because some ribulose bisphosphate is wasted. How is that wasted? Because some of the ribulose bisphosphate has combined with oxygen rather than combining with carbon dioxide. But in photosynthesis, what we require, that carbon dioxide should combine with ribulose bisphosphate rather than oxygen with ribulose bisphosphate. So in that way, some ribulose bisphosphate is wasted, so less ribulose bisphosphate is available to combine with carbon dioxide. Okay. And remember that this process of photorespiration, which we have discussed earlier, where oxygen combines with ribulose bisphosphate, it occurs more readily in two conditions: when there is a high temperature and there is a high light intensity. And you know that both of these conditions, they are found at low altitudes in the tropical parts of the world. Okay. So photorespiration can occur more rapidly at high temperature and high light intensity. Okay, now we have to see what are C4 plants going to do then. to avoid photo, because actually what has to be avoided here, photo respiration has to be avoided, because if photo respiration is not avoided, less photosynthesis will take place. So a plant strategy is to increase the rate of photosynthesis and to avoid photo respiration. So we have to see how is it done. Now you see, this is the transfer section of this is the transfer section of a C4 leaf. Now, a C3 is obviously the ordinary, the plants which we are usually discussing, okay? So if you remember the transverse section of a mesophytic leaf, which you have studied in AS, you remember that, obviously we will see the differences over here. Obviously in those C3 leaves, upper epidermis is there, lower epidermis is there. In the lower epidermis, mostly we see the guard cell, and then you see there are one or two or the three layers of palisade mesophyll cells, and then there are spongy mesophyll cells, and then there is a vascular bundle over there. But what is difference in a C4 leaf? You will see around the vascular bundle there is a ring of tightly packed cells. These tightly packed cells are called bundle sheath cells, right? Remember that they are and then you see, I will come to this point later. Then you see around the ring of this bundle sheath cell there is another ring of mesophyll cells. Okay, they are also tightly packed with each other, and they are also tightly packed with these bundle sheath cells. So it means bundle sheath cells, they have no direct contact with the air which is present inside the leaf. I hope this point will be clear. As bundle sheath cells, they are tightly packed with each other. They are also tightly packed with the mesophyll cells, and in turn, the mesophyll cells, which are arranged in the form of a ring around the bundle sheath cells, they are also tightly packed with each other. So the mesophyll cells, they have a direct contact with the air present. Obviously, air is present here, so it can diffuse inside. So mesophyll cells, they have a direct contact with the air in the leaf, but bundle sheath cells, they don't have a direct contact with the air present inside the leaf. Okay, so this is, you see, this is how this is difference. In case of a C3 leaf, mesophyll cells, they are arranged not in the form of a ring, and secondly there are no bundle sheath cells. Okay, now we come to this, that what are the adaptations of leaf in C4 plants. Now tropical grasses, as I said earlier, that tropical grasses actually, because we said that the process of photorespiration occurs more rapidly in under two conditions: high light intensity and obviously high temperature, and both these conditions are found in the tropical parts of the world. So the tropical grasses, they have evolved the method of avoiding photorespiration. And now how? Obviously we can understand that in photorespiration, ribulose bisphosphate combines with oxygen, and this reaction is catalyzed by rubisco. Okay? So one strategy is this, that rubisco and ribulose bisphosphate, both, they are kept away from high concentration of oxygen. Obviously if they are kept away from high concentration of oxygen, how is the oxygen going to combine with ribulose bisphosphate in the presence of rubisco even, right? So for this, arrangement of cells in the leaf of C4 plant is different from C3 plant, as we have discussed earlier. Around the vascular bundle, there is a tight ring of bundle sheath cells. This ring further surrounded by tightly fitting mesophyll cells. Okay, these 2 points I've already discussed. Now we have to see what happens here. That bundle sheath cells, as I said, they have no direct contact with air, right? And bundle sheath cells, remember that they contain ribulose bisphosphate and rubisco now. ठीक है, इसलिए जो लाइट क्योंकि जो C3 लीफ है, अब C3 प्लांट्स में क्या होता है, we know that the process of photosynthesis takes place inside the chloroplast. ठीक है, and we know that कि chloroplast सबसे ज़्यादा कहाँ पे होते हैं, palisade mesophyll cells में, और उसके बाद spongy mesophyll cells में. ठीक है, अब उसमें क्या होता है कि एक ही क्लोरोप्लास्ट के अंदर आपकी लाइट डिपेंडेंट स्टेज भी हो रही है और लाइट इंडिपेंडेंट स्टेज भी हो रही है। बट यहाँ पे आके अब क्या मसला हो जाता है कि जो आपके मीजोफिल सेल्स हैं, they don't have ribulose bisphosphate and ribisco. It's only bundle sheath cells which contain ribulose bisphosphate and ribisco, and we know that they don't have any direct, these bundle sheath cells, they have no direct contact with the air present inside the leaf. ठीक है, अब क्या होगा, obviously we know that mesophyll cells, they have direct contact with the air, so carbon dioxide in air is absorbed by the mesophyll cells, right? No, so this arrangement ensures that bundle sheath cells are isolated from air inside the leaf, okay? So photorespiration is prevented by preventing oxygen reaching bundle sheath cells, because earlier we discussed a point that ribulose bisphosphate and ribisco both are kept away from the high concentration of oxygen. अब obviously air जो है, it's not only carbon dioxide जो diffuse करती है mesophyll cells में, air obviously oxygen भी present है, वो भी diffuse करती है, लेकिन bundle sheath cells तक अगर oxygen पहुंचती भी है तो वो बहुत low concentration होती है, ठीक है? So that doesn't matter much as compared to that if ribisco and the ribulose bisphosphate, they were present in the mesophyll cells, ठीक है? So I hope this point is clear. Now next is, this is just I have showing you a section through a leaf of sugar cane. As you can see over here, in case of sugar cane is once again you know, C4 plant. There is, we can see over here, these are the vascular bundles, then there is a ring of bundle sheath cells, and these are the mesophyll cells. And this is a section through the leaf of the maize, which is once again a C4 plant. This is not very clear, but this is very clearly given in the book. So now we come to the C4 pathway. Obviously C4 pathway फिर हम उसी reference में कहते हैं because in this pathway the very first compound formed is 4 carbon compound, so that's why this is called as C4 pathway. Now you see as we said, now these are the air spaces of the leaf, so air is present over there, carbon dioxide is absorbed. Now this is a mesophyll cell and this is a bundle sheath cell. We have just taken one, as you know, they are tightly joined with each other, okay? So we have taken one mesophyll cell and one bundle sheath cell over here. So carbon dioxide is basically absorbed from the air into the mesophyll cells, okay? Inside the mesophyll cells, now we know that mesophyll cells, they are in contact with the air. Now mesophyll, now carbon dioxide over here, it combines with a 3 carbon compound, 3 carbon molecule, which is called as phosphoenol pyruvate, okay? So when phosphoenol pyruvate which is a 3 carbon molecule, once it combines with carbon dioxide, as a result of that a 4 carbon molecule is formed, which is called as oxaloacetate. Okay, so you can see over here, this is the very first compound formed in the light independent reaction. So that's why these plants are called as C4 plants and this pathway is called as C4 pathway. Then oxaloacetate is further converted into malate. Malate is once again a 4 carbon molecule. Now this malate passes into bundle sheath cell. In bundle sheath cell, from the malate, carbon dioxide gets removed and it is converted into pyruvate. Obviously, carbon dioxide is removed. Once again, it changes into 3 carbon molecule, which is called pyruvate. Okay? Now we see what happens to the pyruvate. Pyruvate again diffuses back into the mesophyll, where it once again going to accept, where it changes into, where it is used to regenerate phosphoenol pyruvate, which is once again ready to accept one carbon dioxide once more. Okay, so this cycle continues, right? So once again I will repeat it. Carbon dioxide is absorbed from the air by the mesophyll cells. Carbon dioxide in the mesophyll cell combines with a 3 carbon molecule, which is called as phosphoenol pyruvate, and as a result a 4 carbon molecule is formed, which is called as oxaloacetate. This oxaloacetate is further converted into a 4 carbon molecule, which is malate. Malate then passes into the bundle sheath cells. From the malate in the bundle sheath cells, carbon dioxide is removed and this changes back into a 3 carbon molecule, which is called pyruvate. Pyruvate passes back into the mesophyll cells, where it is used to regenerate phosphoenol pyruvate, and phosphoenol pyruvate is once again ready to accept one more carbon dioxide. Okay, now remember that over here, I will come to this later on. This combination on this reaction of carbon dioxide with phosphoenol pyruvate, that is catalyzed by an enzyme which is called as phosphoenol pyruvate carboxylase. Okay, phosphoenol pyruvate carboxylase is the enzyme which catalyzes the reaction of carbon dioxide with phosphoenol pyruvate. So now we'll come back to this. Now the carbon dioxide is free over here in the bundle sheet cells once it has been removed from the mallet. So once again carbon dioxide, it enters into the Calvin cycle as in the usual way, and sugars are formed which are going to be transported to the vascular tissue. So this was the flow chart of the C4 pathway. Now we will just look at it now, the summary of this. As mesophyll cells have direct contact with air in the leaf, so carbon dioxide from air combines with the 3 carbon molecule called phosphoenol pyruvate. The resultant molecule formed is oxaloacetate, which is a 4 carbon molecule, hence the name is C4 pathway. Enzyme which catalyzes this reaction is called phosphoenol pyruvate carboxylase. Remember, this enzyme does not carry out oxidation, so no photorespiration. Moreover, phosphoenol pyruvate carboxylase, like this enzyme, that has a greater affinity for carbon dioxide than ribisco-A1. Oxaloacetate formed is converted to malate. Malate passed on to bundle sheath cells. Carbon dioxide removed from malate molecule. So carbon dioxide enters into the Calvin cycle in the usual way, where it after the removal of carbon dioxide from the malate, pyruvate, a 3 carbon molecule is formed. Pyruvate passes back into the mesophyll cells and converted to phosphoenol pyruvate. Now you see, you will might be there a question comes over there that we are saying that the Calvin cycle is operating over here. Basically that pathway is included in the light independent stage where phosphoenol pyruvate is combining with the carbon dioxide. Okay, so that's why we say the very first compound formed, because otherwise in C3 plants it's only the Calvin cycle, but in C4 plants, other than Calvin cycle, another cycle is added there where carbon dioxide is combining with the phosphoenol pyruvate, and in that cycle obviously, that is, as that cycle is a part of light independent stage, so obviously the very first compound formed over there is a 4 carbon compound. Okay, one more thing, enzymes of C3 and C4 plants are also different. Okay, enzymes in C4 plants, they usually have a higher optimum temperature than the C3 enzymes. That is once again an adaptation to grow in hot climates. Okay, because obviously if the enzymes are going to have a lower optimum temperature, and if the plant is growing in a hot climate, that enzyme is going to get denatured. So that's why enzymes in C4 plants, they have a higher optimum temperature, and that this adaptation helps them to grow in the hot climates. For example, in amaranth, which is a C4 plant, phosphoenol pyruvate carboxylase, that has an optimum temperature of around 45 degrees Celsius, and if temperature drops to 15 degrees Celsius, this enzyme loses its about 70, sorry, what's that? So, and if we say that phosphoenol pyruvate carboxylase in case of amaranth, which is a C4 plant, it has an optimum temperature of around 45, and if temperature drops to 15, this enzyme loses its 70% of activity. So you can see activity बहुत कम हो जाती है. But the same enzyme in pea plant, and pea plant is a C3 plant, this enzyme has an optimum temperature of 30. Now see a comparison of 45 and 30, it's a very very low temperature, and can continue to work at a much lower temperature than in amaranth. Okay, so this is how the C3 enzymes are also different from the C4 enzymes. So with this, we almost finish our chapter of photosynthesis. What is just left behind, that is that is actually the experimental part of this chapter, that how we can investigate the rate of photosynthesis using an aquatic plant, and how can we investigate the effect of various factors on the rate of photosynthesis. Okay, um, though the next coming week you will be conveyed about it, that is going to be off for A level students, and then the classes will be resumed, as per the government's announcement on 7th of September. And hopefully we are going to have face to face classes, inshallah. So, but I will try my level best to prepare a presentation on, because that's a very, very small subtopic. But if I was going to include it in today's topic, it was going to be a bit lengthy. So that's why I just skipped it. So I will try my level best to have it uploaded on the YouTube, so that you can view it. And when we resume the classes once again, and inshallah if we are going to have the face to face classes, then we can start with the next chapter rather than just completing a part of that. Okay, I will convey it in the group when I will get it uploaded. Okay, so till then, take care, have a good have a nice long week, off week, and till then, take care of yourself. Allah Hafiz.